Everybody, welcome. Today we're going to have a seminar from Xiang from the Department of Biostats. He did his postdoc at the University of Chicago, Matthew Stevens' group. And he got his master's and PhD at Duke University. And today he's going to tell us about differential expression analysis for RNA-seq data.
Thank you. Thank you, Alan, for the invitation, and thank you all for coming. So today I'm going to talk about some differential expression analysis methods our group is developing. And this main work is led by my talented student, Sichuan Shen. And so, as we all know, that RNA is very important, right? So RNA is mediating the signal from this DNA up to the protein. So, and it's essentially because it's a central role in this biological process. It plays a very important role in, say, like disease etiology, and plays an important role in development. And so therefore, it's important to understand the RNA, the role of RNA, different gene expressions. And to do that, a lot of people nowadays can easily do a lot of RNA sequencing studies or microarray studies. And once you get those results, get this data, the first important step is essentially try to identify those differentially expressed genes that are influenced by predictor of interest. And those predictor of interest can be many different things. So there could be disease status, there could be risk factors, there could be environmental covariates, or in the case of this gene mapping studies, this could also be genotypes. So identifying those differentially expressed genes with respect to this either environmental or genetic covariance is a very important first step to understand the genetic basis of disease susceptibility and also understand the genetic and the environmental basis of phenotypic variation. Now, to identify those differentially expressed genes, the first important step is to do RNA sequencing nowadays. And the RNA sequencing can be very easily summarized in these multiple steps. So to do RNA sequencing, you first basically extract your RNA from your sample, right? And then you do a reverse transcription to get your cDNA so that it's more stabilized. And then you can cut the cDNA into different fragments and construct a library so that you can sequence all those fragments. And once you get a sequence of the fragments, you can try to map it back to the whole genome so that those small short reads will be mapped into those exonic regions. So if a gene is heavily expressed, then you will see a lot of reads mapped to that particular gene. And if a gene is lowly expressed, then you tend to have a much less read maps over there. So as you can see that at the end of the day, the RNA sequencing data will give you a lot of read counts data for every single gene. So therefore, the first important feature of RNA sequencing data is that the raw data, is actually in the form of counts. So because this RNA sequencing data are count-based, and you can see that this gene expression variation across the technical replicates can be usually accurately described by a Poisson distribution. And in addition, once you have biological replicates, and then you usually have other confounding factors or biological noise, then in those cases, gene expression variation across those biological replicates often displays over dispersion. So in order to model those RNA sequencing data, especially once you have biological replicates, sometimes you will have to use more over-dispersed Poisson distribution to model that, which are also commonly known as negative binomial models. So for example, the most widely used methods, as we know it, are HR and DSeq, or the second version of DSeq. And those methods are widely used to analyze, perform differential expression analysis in RNA sequencing data. And the backbone of those methods is essentially a negative binomial model. They try to model those raw counts directly. So although there are also many non-counts based methods, for example, lemma can also be used to analyze RNA sequencing data because the RNA sequencing data itself is a raw count. And because of its over-dispersion often observed in this raw count data, it turns out that it's very useful to model this count directly using this type of negative binomial models in order to get more power and control for false discoveries in RNA sequencing data. So therefore, over here, you can see that the first important feature of RNA sequencing data is it's in a count-based form. And because it's in count-based form, it's extremely important to account for this mean and variance dependence through modeling either Poisson or dispersed Poisson models. So that's the first feature. And the first feature is well recognized and used by many different studies. But the second feature I will mention over here, it actually has been largely ignored by all those differentially expressed RNA sequencing methods. And this second feature is that gene expression are actually heritable. So as we all know that we all know that the gene expression levels across the human population, across multiple individuals, they always have a heritability component. They always have a genetic component. And in fact, if you perform a relatively large population study and try to see how much proportional variance in gene expression level can be estimated by genetic basis, or to estimate this so-called gene expression heritability, you can see that those gene heritability are relatively large. So the average gene expression heritability, say in the peripheral blood, is estimated to be 15% to 34%, and depending on which data source you collect, which sample size you collect. And also in other tissues, say adipose tissues, the average heritability is also estimated to be relatively high, so it's 23%. And in fact, across all those genes, you can see that the highest heritability estimates in all those tissues can be high as 90%. So therefore, there's a huge genetic component. And there are many recent genetic mapping studies try to identify those genetic components, and they have identified thousands of cis-eQTLs. So those are the SNPs that nearby the genes that influence in the gene expression level. So the heritability can be easily explained by those cis components. And besides the cis components, there's also a large remaining trans component that still remains unidentifiable. So for typical gene heritability, in fact, 90% of them can be still, only 10% of them are explained by cis SNPs, while 90% of them remain to be discovered, and those are explained by trans components. So therefore, those gene expression levels have very large genetic basis. And because gene expression levels are heritable, so you can see that the gene expression will typically co-vary with the kinship or population structure. So if you collect a data set that contains related individuals, and because these individuals are genetically related, so that, and because the gene expression level are heritable, so those genetically related individuals will also have a similar gene expression levels. So the genetic relatedness will cause this similarity in gene expression levels. So therefore, it's important to account for those individual relatedness or population structure we typically account in this data. And we all know that in genome-wide association studies, it's important to account for those type of structure, whether it be its family structure or whether it's a population structure. And if you fail to do that, then you are going to have a huge type 1 error, and you are going to have a lot of spurious associations or even reduce the power. And certainly, gene heritability is not the only reason that can cause gene covariance sample non-independence. So in fact, there are other forms of confounding effects. For example, batch effects then can also cause sample non-independence. So for example, if you process the data set, those are consists of completely unrelated individuals. So individuals are indeed unrelated. But if you happen to process this data, say there's 2 individuals in the same batch, then because of the batch effect, the expression level on many different genes between these 2 individuals are also going to show a similarity between these 2 individuals. So therefore, you can see that individual relatedness, population structure, and those hidden confounding and batch effects can all cause sample non-relatedness. And it is actually extremely difficult to account for a sample non-relatedness. And so when you have a data, RNA sequencing data, that has the first feature that is count-based, and also has a second feature that gene expression are heritable, and you have the sample non-independence, then you definitely want to account for both 2 different features. But those previous approaches have only been focused on one of those 2 features. So for example, one approach, they try to normalize the counts to continuous values. And then once they can normalize the count to continuous values, then they can use the linear mixed effects model to account for population structure, individual relatedness, or population stratification. And in this approach, while they deal with this population structure issue, they actually fail to account for the count-based nature of RNA-seq. So naturally, you would expect that this type of approach may lose you power. On the other hand, there's a second alternative approach. And this second alternative approach is essentially to use the negative binomial-based models like DSeq and HR to analyze your RNA sequencing data. But this approach fails to account for this individual relatedness and sample non-independence in the data. So intuitively, once you do have those related individuals, or once you do have those hidden confounding effects, which are actually quite common in the sequencing studies, then you would expect you are going to have more spurious associations or lose of power. So therefore, you can see that neither of those previous approaches are satisfactory in dealing with these two important features of RNA sequencing. So motivated by this observation, then we decided to develop a new statistical methods called Poisson mixed effects model, try to account for both features of this, both this type of features observed in the data. So basically, well, that looks ugly actually. I don't know what's going on. I probably made this slide in the Mac, and this is the Windows PC. So basically, the mixed model does essentially look at each gene at a time. So we are going to do a differential expression analysis, and to do that, we basically look at each gene at a time and fit this model. And this model looks actually quite straightforward. So we're going to model this count data, which is the Y subscript I. So this is the count data for I's individual. And because it's count data, we naturally model it as a Poisson distributed. And so with the Poisson distributed with the rate parameter that depends on these two factors. One factor is the total read depth. So intuitively, if you sequence and get a more high coverage for that individual, then you are going to have a more read count for that particular gene, right? So therefore, it's scaled with n sub I. And then in addition to this n sub I, we're also going to have this lambda sub I, that's the unknown rate parameter. And so therefore, yi follows a Poisson distribution with rate parameter that's a product of n sub I, this read depth, and another unknown parameter, lambda sub I. And then we are going to model this lambda sub I as a function of the predictor of interest. So because this lambda I is a value between 0 and infinity, so we take the log transformation and we model it as a linear function of many covariance effects and a predictor of interest. So over here, we have our log lambda sub I, and then we have a W sub I times our alpha. So the W sub I is essentially all those covariates you want to include in, which also include the intercept you want to include in, while the alpha is the effect size. And besides this, standard covariates you want to fix, you want to control, for example, the covariates can include age effects or sex effects you want to control for. And besides this, you also are going to have your predictor of interest. So this X is our predictor of interest. So this predictor of interest can be anything, can be any continuous or discrete values. So for example, if you are interested in identifying and say cis QTL, then this X is essentially your genotype vector as coded as 0, 1, 2 as copies of your reference allele. Alternatively, if you're interested in, say detecting those age-associated or gender-associated gene expression levels, then X could be an age or X could be a gender level, 0 or 1. So the X is our predictor of interest and the beta is the coefficient of interest. So our goal is really to estimate the beta effect size and test it to see whether it's equal to 0 or not in order to identify differentially expressed. So these two are very natural components and standard components in Poisson mixed effects models. But in addition to these two, we're going to have two extra terms. So we have this G term, G sub I term, and we also have this E sub I term. So this G sub I is the genetic effect for I's individual, while the E sub I is the environmental effect for I's individual. And our novel part of this model is essentially introducing both the G sub I and the E sub I to account for those genetic and environmental effects. So intuitively, those are the fixed effects. W times alpha, X times beta, those are fixed effects, while we will model those G and E as random effects. And because they are random, they are going to introduce more variation than you would expect under the Poisson model. So both of those terms are going to deal with this overdispersion issue that's commonly observed in RNA sequencing. So, although both of G and E, they model over dispersion, the variance not accounted for by the standard Poisson distribution, they actually model two different aspects of over dispersion. So in particular, the E models the variation that's due to independent environmental noise. So if you combine all those n values of E together, right? So now the E, the capitalized E is the n by 1 vector of environmental effects across n individuals. Now this will follow some multivariate normal distribution with the variance equal to sigma squared times 1 minus h squared. So basically the variance, those E's are assumed to be independently and identically distributed with the normal with the variance equal to sigma squared times 1 minus h squared. So therefore the E that models the standard independent variation. So intuitively, if you only have this E term, then this looks very much like a negative binomial model, except the functional form is slightly different. So you can think of our model with only this E term is equivalent to this negative binomial model that people are always using. But in addition to this E term, we also have our G term. Now the G term is the key factor here. Try to model the sample non-independence due to either individual relatedness, population structure, or hidden confounding effect. So this G term here is, again, the G is an n by 1 vector. And we model this n by 1 vector as follow a multivariate normal distribution, but this time our covariance structure is more complicated. So instead of assuming all those EIs are identically distributed, now we have those GIs that are actually correlated depending on the structure of this K. So the K matrix is essentially try to capture the sample non-independence. So intuitively, if I and Jesse individual are genetically related, or I and Jesse individual that come from the same batch, then this K sub ij is going to be large and positive. And when K sub ij is large and positive, then it will introduce this similarity in this G sub I and G sub j. So that G sub I and G sub j will be similar to each other. And therefore, by modeling that, by controlling for this G, you're essentially controlling for the similarity between I and Jesse individual. So therefore, you essentially controls for the sample non-independence you commonly see in RNA sequencing studies. And so therefore, this G is a unique feature of our model that models the sample non-independence due to individual relatedness of population structure. And over here, we'll assume that the K is known a priori. So if you have genotype data, it's quite easy to compute a kinship matrix, right? Or if you have pedigree data, you can also compute the kinship matrix. And alternatively, if you only have gene expression data, and if you want to control for hidden confounding effects, and because the gene expression data actually contains a lot of information on where the confounding effects are, you can directly use the gene expression matrix to compute the gene expression covariance matrix as a K matrix of cut here. So therefore, the K is essentially a pre-computed matrix that models an individual non-independence. And finally, over here, we can see that we have two factors, the h1 minus h square and also h square. So if you do have genetic data, then this h square is essentially your heritability. So in fact, you can also use this model to estimate the heritability for every single gene. So this is our model. So you can see that compared with other models, other previous negative binomial model like HR and DSeq, our key feature is we have this extra G term to account for individual relatedness. So intuitively, once you do have individual relatedness, sample non-independence in your data, then you would expect our model to work relatively well. So this model is straightforward and intuitively makes sense, but to fit this model is actually not. Fitting this model is actually quite difficult. And that's because it turns out the model we're looking at belongs to this Poisson mixed, it's called the Poisson mixed model that belongs to the generalized linear mixed model family, which is commonly referred to as GLMM. And the inference in this GLMM family is very difficult because we have an n-dimensional integration that cannot be solved analytically. And over here, the integral is actually n-dimensional, so it's the sample depends on the sample size. So even if once you have an even larger sample size, it actually gets more difficult to do the inference in contrast to other methods. Once you have a larger sample size, it usually makes sense easier. So, and there are some standard frequencies approaches we can use, right? So that's include numerical integration that's based on Gauss-Hermite quadrature and all Laplace approximations. But neither of those approaches actually scales well with the dimensionality of this integral, because once you have a huge dimension, then neither numerical integration or Laplace approximation is going to give you accurate answer. So therefore, it turns out that those commonly used methods, such as the PQL, often fails to account for model uncertainty and can give you biased estimates. So it gives you biased, not only biased beta estimates, but also biased standard error of betas. So therefore, we decided to take an alternative approach. And this alternative approach is to do a Markov chain Monte Carlo or MCMC-based methods. And the nice feature of this MCMC-based methods is that it has naturally accounts for this uncertainty in all those hyperparameters because it's actually drawing chain. And in fact, if you run this chain over and over long enough, then you are guaranteed to get into the stationary distribution. So therefore, that's extremely beneficial of this MCMC type approach. However, the MCMC type approach also has a common known issue, and that's its computational burden. So it's extremely computational heavy. So in fact, every iteration of this MCMC approach scales cubically with the sample size. So that makes inference extremely difficult. So therefore, we decide to use MCMC, but we make several novel algebra innovations to reduce the computational burden we have. So in fact, we decide to use to take advantage of two algebra tricks. One trick is based on this recent auxiliary variable-based MCMC algorithm. So essentially, develop auxiliary variable representation of this Poisson model so that you no longer need to sample the count data, but rather sample the continuous data. And this makes things relatively easy. And I'll get to this point in more details later, shortly. And the second trick we use is the recent mixed model innovations. So essentially, this type of innovations applies once you have a kinship matrix. And essentially, this allows you to do eigen decomposition at the beginning. And then at the later steps, later MCMC steps, you no longer need to do those cubic operations. And so therefore, combining by both, our methods can actually reduce the per MCMC iteration complexity from cubic down to quadratic. So essentially, it gives you an n times faster algorithm. And n is the sample size. So if you have a larger sample size, you can see that the speed gain you are going to have is much more dramatic than if you have a much smaller sample size. And once we have this efficient MCMC approach, then we actually goes back to our frequentist approach. So instead of doing a Bayesian inference, we actually based on asymptotics and use posterior samples to obtain approximate maximum likelihood estimates, and also its standard errors. And with that, we can convert them into a p-value, and we'll show you later that this approach actually gives you well-behaved p-values. So that's our main idea of doing inference on this Poisson mixed effect model. And we call our algorithm the Markov algorithm, which stands for the mixed model association for count data by data augmentation, because one key feature is using this data augmentation approach. So this data augmentation approach is actually relatively straightforward. So basically, right, we have a count data, and we have a count data, we have observed Y counts. Now, observing Y counts in an RNA sequencing data, that's also equivalent to imagine a Poisson process on this linear line, right? So if you imagine a Poisson process in this line, and this event will happen randomly on this particular line based on this Poisson process. And observing Y counts in, let's say, time interval between 0 and 1, right? Observing Y counts of RNA sequencing rates, that's equivalent to observing Y counts in this time interval of 0 and 1 of this Poisson process. And that's equivalent to say that your Y's count happens before t equals to 1, right? And plus your Y plus 1's count has to happen after t equals to 1. So therefore, all you need to do is to just record the time of Y's counts and record time of Y plus 1's counts and make sure that t equals to 1 happens between these two. And this will give you equivalent measurements on the Y's counts in this interval. So therefore, in fact, instead of observing Y's counts, we essentially can say that we observe our Y's counts in an interval at t sub 1 that happens before t equals to 1. And also, the inter-arrival time between Y's count and Y plus 1's count equals to t sub 2, and this t sub 2 includes the t equals to 1 time points, right? So therefore, instead of directly modeling this Y's count, we can just model this arrival time of tau sub 1 and the inter-arrival time of tau sub 2. And both tau sub 1 and tau sub 2 are actually continuous data. So therefore, instead of dealing with very difficult Poisson likelihood, dealing with this very difficult count data, now we can turn this count data into a continuous data, and we can perform our sampling on this continuous data, which makes things much, much easier. So that's the main trick of this data augmentation approach. And because of this data augmentation approach we use, and also because of some other novel algebra tricks we use for mixed effects models, we can see that our MCMC is extremely fast. So compared with the standard MCMC methods, which is called the MCMC GLMM method, which is this orange line, if we record the computation time, and also this, and also at the log scale, you can see that our method, called the purple line, works much, much faster. And this difference is much obvious once you have an even larger data set. So if you have 1,000 individuals, then our method is roughly about 1,000 times faster than this competing methods. So therefore, you can see that now this new computational method allows us to perform MCMC on this extremely difficult Poisson mixed model. And because that, this allows us to actually apply this Poisson mixed effects model to relatively large scale RNA sequencing studies quite easily. Now, after this MCMC computation, what we do next is to compute the p-values. So again, we're not interested in doing Bayesian inference, but rather we want to use the MCMC as a machinery to get the p-values directly. And to do that, we essentially rely on this asymptotic normality of both the likelihood and the posterior. So we all know that our posterior equals to the likelihood times the prime, right? So if we want to make inference on the likelihood, then all we need to do is to compute likelihood as a ratio between your posterior and your prime. So because you already know your prime, and you also get your MCMC sample and get your posterior, so therefore simply take the ratio between these two gives you your likelihood function. And so more specifically, we essentially will assume that the prime distribution beta follows the normal distribution 0 and sigma square 0, 1. In fact, it actually doesn't matter because the prime inference will be canceled out from this posterior to give you likelihood. And then we compute the posterior, and then we use the method of moments, try to match the first and second moments, and eventually we get back to our beta hat estimates. So this beta hat estimates is based on our posterior estimates of beta and the prime estimates of beta. This beta hat estimate is actually a maximum likelihood estimate. And in addition to that, we can also easily compute the standard error for those MLE estimates for beta. So therefore, from this MCMC algorithm, we eventually get our beta hat and the standard error of beta hat, and that gives you a p-value directly. So you can take the ratio between the two, that's just t-statistics, and you can easily convert it to a p-value. Now you probably wonder that this is MCMC-based approach. You probably never heard of or never saw this type of approach before. So one naturally wonder whether this works at all or whether this is stable at all, because we all know that MCMC depends largely on the initial value. So it turns out that the results are actually quite stable across multiple runs. So if we basically compute we basically did 3 simulation studies with the heritability equal to either 0 or 0.3 or 0.6, which I'll get to that in more details and shortly. So in those 3 set of simulations, we run 2 different runs of Macau. So we starting with a different set of initial value for run 1 and the different set of initial value for run 2. And you can compute their minus log 10 p-values and correspond to each other. So you can see that the p-values from this one set of MCMC chains looks very similar to the second set of MCMC chains, suggesting that our MCMC-based p-value actually is extremely stable across multiple runs. So now you can see that we now have this very nice Poisson mixed effects model. And this novel part of this model is try to account for the sample non-independence. And we also have this very efficient MCMC-based p-value computational algorithm that actually makes it eventually scalable to large-scale RNA sequencing datasets. So now we want to see that whether our approach is indeed behave as we would expect. So to do that, we first did a null simulation. And so we simulated 10,000 genes. And we also extract the kinship matrix from a Baboon data, which I describe later on. And so this is essentially a 63. So this is the RNA sequencing data done by a wild Baboon population, and that consists of 63 Baboon individuals. And so this kinship is 63 by 63 K-shaped matrix. And then we simulated this bunch of different settings, and so that all those settings, they have relatively either small, actually either 0 or moderate or large genetic effects. So we fixed the heritability equal to 2.3, or we also checked whether it changes to when h squared equal to 0 or 0.6. And besides that, we also make sure that the predicted variable x is correlated with the kinship matrix, and so that it also has the heritability equal to either 0 or 0.35 or 0.75. And then we compare a bunch of different methods. So those methods include those. So the first one is the linear regression. So what we do is essentially we just normalize the RNA sequencing data and fit a linear model to that and see and to compute the p-value. A second method we compare is the linear mixed model, and that's implemented in the JAMA software package. So the linear mixed model also normalizes RNA sequencing data first, but it contains this kinship matrix. It contains a random effect, so it can control for individual relatedness and population structure. However, it cannot account for the count nature of RNA sequencing data. And the third method we look at is the Poisson distribution. So as I described earlier, if you only have technical replicates, Poisson is a very good model to describe the distribution. But if you have biological replicates, then you want to use this negative binomial model, which have over-dispersed components. And similar to the negative binomial model, we also compared with the HR and DSeq, which essentially put more constraints on this negative binomial model to make them more specialized. So intuitively, if you have a large data set, then you would expect DSeq and HR to perform quite poorly. And if you have a small data set, maybe you would expect DSeq2 and HR to perform slightly better than negative binomial models. And then besides all this, we also have our Poisson mixed effects model that's implemented in the Markov package. So those are the 7 methods we compared with. And we first compare them in this null simulation. So basically, there's no differentially expressed genes at all. And so in this null distribution, because this is a null simulation, the p-values from the methods are expected to follow a null distribution, which is a uniform distribution. So therefore, if you plot the p-values, the minus log 10 p-values on the y-axis, and plot it against this expected uniform distribution, then if a method does controls for sample non-independence, say individual relatedness or population structure, and then you would expect this line on this diagonal, all those dots line on this diagonal line. So therefore, you can see that among these 5 different methods we look at, we first look at the 5 because we are simulating X is a continuous and DSeq 2 and the HR cannot handle continuous data. And you can see that among those 5 different methods, there's 2 of them give you inflated type 1 error. So in particular, if we look at a Poisson model, right? So you can see a huge inflation of p-values, suggesting you are going to see a lot of spurious association. And the similar thing happens for the negative binomial model, although to a much smaller extent. And in contrast to these models, you can see our method, Markov, and also the linear version, the linear mixed model, JAMA, all controls for type 1 error pretty well. And you can look, if you look at this genomic control factor, the genomic control factors all looks close to 1. So these are the results for these 5 different methods. And again, DCK and HR cannot apply to continuous X values. So in order to see their behavior, we basically have to binarize those X values. And we discretize the values so that we compare adding this HR results and DSeq results. And as you would expect, you can see that this HR and DSeq, they all have inflated type 1 error. And similar to this negative binomial model you see earlier. And this is consistent with the fact that HR and DSeq are indeed based on negative binomial models. So, and this inflation of p-values, it turns out that it actually depends on the sample size. So if you have a small sample size, it's usually hard to see this inflation of p-values. But if you have a relatively large sample size, then you're going to see a much obvious inflation of p-values. So to see that, we basically did a null simulations again, but now varying the sample size on the y-axis, on the x-axis. So now we simulated from a small study, say 60-some individuals, up to a relatively large study with 1,000 individuals. And we basically plotting their genomic control factor on the y-axis. So you can see that when heritability, when hx squared equal to relatively 0, then all of those, only this negative binomial model have a slight inflation of type 1 error, while other ones work relatively well. But once you have a relatively large h square x, then you can see that both a negative binomial and a linear model are going to show severe infection of type 1 error, while our method and also JAMA still remains the same, remains control for the genomic control factors. So you can see that now in the simulation study, we basically can see that both our model and this linear version, the linear mixed model implemented JAMA, can control for sample non-independence. So how about power comparisons now? So one advantage of our method compared with the JAMA, while both they control for type 1 error well once you have related individual, our method can actually model this count data directly. And intuitively, by modeling this count directly, you would expect it to get more power compared with other approaches. So to show this, we basically did this power comparison. And we simulated the alternative set of data. We simulated 10,000 genes, among which there's 1,000 genes that are differentially expressed, and then 9,000 genes are not differentially expressed. And we're varying actually a lot of different parameters to, we compared our methods on this on this set of parameters, but then we vary each parameter one at a time to see which parameter is more influential on the relative power comparison among different methods. And so we again apply for this 7 methods over here and to see which one works better. So here, it turns out that once we vary in different separate parameters, there are 2 parameters that are extremely important in determining the relative power of different methods. So the first parameter is H squared. So H squared is heritability, gene heritability. So if the gene is not heritable, so if the heritability is exactly 0, then we would expect our model actually to perform slightly worse than this negative binomial model, simply because we have one more parameter to model, and simply because the data is actually favoring this negative binomial model. So indeed, you can see the negative binomial works slightly better, although the difference is actually quite small. But now if you have a moderate heritable gene, say the heritability equals to 30%, or highly heritable gene, say the heritability is equal to 60%, now you can see that by accounting for those gene heritability and sample non-independence, now our method can get more power compared with all the other approaches. And in fact, similarly, because JAMA also accounts for sample similarities, JAMA method also performs well than others. But if you compare our method with JAMA, because our method accounts for the count nature of data, so we have a slightly better power than the JAMA method. So therefore, the first factor is the heritability of gene expression. On the other hand, the second important factor that affects the relative power is this coefficient of variation, which essentially is the read depth variation across different samples. Now, we know that if you do RNA sequencing data, say if you give 8 samples in a run, those 8 samples are going to have a different read depth. So although you try to control it on average, say 10 million, they are going to have a variation on this 10 million read. And so the CV is essentially trying to quantify this variation. So intuitively, if every sample has the same identical read depths, then modeling counts don't really have much advantage. But on the other hand, if the CV is relatively high, so if you do have some variation across different samples, which we do see in real data, then we would expect modeling counts are going to get more power compared with not modeling counts. So this is essentially what we see. So if you increase the CV, although all the methods start to perform poorly because it makes them more difficult, you can see that the power difference between our method versus other non-count-based methods, especially JAMA, that becomes larger. So if you don't have much variation, then our method and JAMA looks relatively similar with only a small slight advantage. But now with the CV equals to 0.9, the difference becomes larger. And in fact, the performance of JAMA drops quickly once you have a larger variation of read depth across different samples. So therefore, you can see that the power really affected by these 2 important factors, heritability of genes and the coefficient of variation of this read depth variation. Now, here again, because we simulated X as a continuous trait, so we cannot apply this DSEQ and HR. So we have to discretize this into a 0 or 1 trait, and to apply DSEQ and HR. So as you would expect, that the HR and DSEQ don't really perform well compared to all the other methods, and intuitively that's simply because we have a relatively large sample. So we have 60-some individuals, and for those relatively large sample size, DSEQ and HR won't perform well because they are restricting this negative binomial model. So now these are the simulations we did now and did the alternative. So now let's look at data sets. So we applied our method and compared with other methods in 3 different data sets. And these 3 different data sets actually represent a different situation. So the first data represents the related individual. The second data represents the population structure, while the third data contains the hidden confounding effects and other batch effects. So we can see that our method can be applied to a wide variety of data types. So the first data is this Baboon study. So it's a data set collected by Professor Jenny Tang from Duke University. So basically, so this data is collected on this baboon population in Africa, and she went there every summer and studied this baboon population actually for quite a while. And this baboon population had been established since the early 1970s, and people have been tracing this baboon population for many different generations. And so they collected their behavioral data, they collected their blood types, and they collected all their, and they draw their detailed pedigrees. So there's a lot of information available in this data. But the missing information in this particular data is the genotype. So therefore, for this particular study, they decided to do a genotype, to perform a genetic study, and by analyzing 63 individuals and performing RNA sequencing on those individuals. So this data are collected on whole blood from these baboons. And we have 63 individuals, and we have about 12,000 different genes. And among those baboons, although we have a lot of different covariates, we decided to focus on the variable sex, simply because it's easier to see whether the detected sex-associated genes are true or not, because intuitively, sex-associated genes should be enriched on X and Y chromosomes, right? And so in this particular data, this feature of this data is that they contain these related individuals. So in fact, the 63 individuals are collected from relatively large pedigrees, so they are more or less related to each other. And to estimate this relatedness, to estimate this kinship matrix, we use the microsatellite data collected on this baboon population to estimate the K, so we can plug in the K matrix and run our macro analysis. So you can see, so here shows the results. So on the left side, it basically shows the power versus force discovery rate across a bunch of different methods. On the right-hand side, I also show the enrichment of X chromosome genes. And so that's as an indicator on whether a method works better than the others. So in either case, if a method works better than others, then the line should be higher than others. So you can see clearly our method, macro works better than all the other methods. And in fact, if we just focus on the force discovery rate, for example, at 10%, yeah, our method actually gives you 50% more power compared with the next best approach. That's a negative binomial model. And compared with the standard approach, say HR and DSeq, those standard approaches are sitting over here, consistent with our simulations, right? So it doesn't really give you much power if you use HR and DSeq to analyze your data. So this is the Baboon data. The second data we look at is the fusion data. So this fusion data is collected. So it's a study of Finland and the United States investigation of this NIDDM genetics. So they basically collect a relatively large RNA sequencing study with skeletal muscles, and the data is collected on 267 individuals, and they measure 21,000 genes. And in this data, we are primarily focused on 2 variables that are particular of interest. So the first one is the OGTT test, or oral glucose tolerance test. So this is essentially an indicator on whether the individual is a type 2 diabetes or not. So this is a binary indicator. And besides this type 2 diabetes status, we also look at this glucose level, which is a continuous trace. And in this particular data, we have this population structure because they're collecting data from Finland. And this data is actually collected samples from 3 different towns in Finland. So if you simply do a principal component analysis, you would see that PC1 and PC2 capture the 3 different town structure in this Finland. And in order to control for population structure, as usual, we use the genotype data to estimate relatedness matrix K, and we plug in so we can apply the Markov tool. And so we did a study compared with a bunch of other methods, and the top 2 panels basically shows the results for type 2 diabetes status, while the bottom 2 panels shows the glucose levels. So the first column shows, again, the power versus the force discovery rate. And while the second column is different. So in the first second column, we essentially to look at whether the top T2D identified T2D associated genes, whether they have a high overlap or not with this top GL associated genes. So intuitively, this glucose levels and the type 2 diabetes, they are really biologically relevant traits. So by looking at whether the method gives you a similar answer on those two associated genes, then you would basically say that which method works better. So if the method works better, then you would expect to gives you a higher overlap. So again, among those different methods, you can see our method Markov works better than all the other methods. And in fact, if you look at this GL-associated genes, right? So at 10% FDI, if you use the other methods, you simply won't detect anything. While if you use our Markov method, that actually gives you 100 signal. And if you look at the overlap, you can again see a very similar trend that our method works better than others. Now over here, this T2D, this is a binary trait. This glucose levels is actually not a binary trait. So it's and so in order to apply DSeq and HR to this type of data, we have to discretize this glucose levels into 0 and 1 based on the median levels. But discretize this glucose levels to 0 and 1 actually loses you a lot of power. So in fact, we no longer detect any GL-associated genes at all, and even at a relatively flexible FDR cutoff, say 20%. However, we can still do an overlap between this top GL01 associated genes with the top T2G associated genes, or with the top T2D associated genes with the GL01 associated genes. So essentially doing this two reciprocal enrichment analysis. So you can see again, our method works better than others. And if you look at HR and DSeq, again, they are the worst methods among all the 7 methods. And this also intuitively makes sense. And finally, all those 2 previous examples, I focused on relatively related individuals and the relatively large data. So the final data, final analysis, we decide to focus on unrelated individual and relatively small data. So to do that, we look at this Yoruba and RNA sequencing data that's performed on 69 Yoruba individuals who are a part of this HEPMAP population. And we get this data, RNA-seq data, and this RNA sequencing data measured on gene expression levels on 13,000 genes. And this is still relatively large data. So in order to see our performance in relatively small data, we decide to do a subsampling. So we basically created 6 samples, or 10 samples, or 14 samples by random sampling from this 69 individuals. And we repeated this many times so we can average the power across this different subsampling data sets so you can get a more robust estimate of power difference between these various methods. And in this data, we again look at this sex as a predictor variable of interest, and simply because it's easier to see to compare the methods, we can use the enrichment of sex chromosomes as an indicator to do that. And in this particular data, as mentioned, they are all unrelated individuals. There's not really much population structure going on, but we do have some hidden confounding effects. And we all know that hidden confounding effect is a huge problem in all sequencing studies, whether that's in RNA sequencing or other bioscientific sequencing, for example, studies. They're always going to see this hidden confounding effects, and it's always important to control for that. So therefore, we are using this data to show how our method can also use to control for hidden confounding effects, so get a better performance compared with other methods. And in order to control for this hidden confounding effects, we basically use the gene expression data to construct the covariance matrix. So intuitively, if the 2 samples are placed in the same batch, then many of the genes will have similar expression levels. So therefore, by computing this covariance matrix, then the corresponding covariance matrix would be able to capture those hidden confounders. And so here are the results. So I'm listing the results for n equal to 6 case, n equal to 10 case, and also n equal to 14 case, 3 different, very small, relatively small data sets. And then we use the enrichment of X chromosome genes as an indicator to evaluate the method's performance. And this is simply because in this data, we're focused on very small samples, and you actually don't have much power to identify it if you look at the traditional power versus FDR power. So in this enrichment, again, you can see that our method works pretty well when sample size equals to 10, also equals to 14, and works similarly either as say a Poisson model or negative binomial model, depends on which region you look at, when the sample size is very small, equal to 6. And among all those comparisons, if you look at the standard methods, say DSeq and HI, and you can see that considered with all the previous data, this methods also then perform really very well, even in those relatively small data sets. So you can see that in the third data, by controlling for those confounding factors, our method also performs better than others. So finally, I'm just going to list the computation time. So one key downside of our method is that it is computationally slow. So if you just look at using the other methods, say using GMMA to fit a linear mixed model, that only takes actually less than a minute to analyze the baboon data and about a minute to analyze the human data. But if you decide to use the Markov method to do that, then you are going to use an hour for the baboon data and almost a day for the human data. So you can see that it's a huge computational cost. However, even though our computational cost is high, ours is much faster than the comparative algorithm, MCMC or MM, right? So it's about 10 times faster here and 100 times faster there. And in fact, even an hour or 1 day is relatively small cost comparing with the time you have to spend on collecting the data on generating our RNA samples. So finally, as a quick summary, so we basically presented you a method called a Poisson mixed model, and also presented a very efficient Markov algorithm to fit this type of models. And this methods are extremely important to dealing with RNA sequencing data when you have sample non-independence. So the sample non-independence can come from many different ways. So it can come from individual relatedness, can come from population stratification or structure, or it can also come from when you have hidden confounding effects in your RNA sequencing data. And by controlling for sample non-independence, you can get more power, and you can also get a proper control for type 1 error. So, and the manuscript now is on bioRxiv, and you can also download the software Markov from my website. And if you are interested in using the R package, just let me know. We also have an R package to do that. So finally, I would like to thank especially Sichuan, and he is really a talented student, and he pioneered all the work on this study. And I also want to thank Michelle and Laura, who are collaborators on the study over here, and also Qingke, Sheyang, and Jenny, they are also collaborators on the study. And I especially want to thank Mike Banke for providing this fusion on the sequencing data too. And finally, thank you all, and I'm happy to answer any questions.
Questions?
So like you mentioned earlier in your the simulation study with the Baboon data, so you choose a heritability of like the H square is 0.3,
Yeah.
and then the kinship matrix you use 0, like 0.35 and 0.75.
Right.
I'm wondering, like in real life data, how do you determine these values?
So that's a very good point. So over here in the simulation, we basically try to make the simulation as real as possible, and that's why we use the Baboon data to do that. And so we essentially use the same kinship matrix as in the Baboon data. So the K matrix is identical as the Baboon data. And so that's realistic. And second, we also use the H square. So as you mentioned, we value 3 different values, 0, 0.3, and 0.6. And that's because the median heritability of gene expression is around 30%. So that's why we use a 0.3. And then 0.6 is roughly the 90th percentile, and 0 is the lowest value you can get. And in fact, for all the other data, say the sigma squared over dispersion variance, we also try to estimate from the real data and do the simulation. So we actually try to estimate all the parameters from the real data to make the simulations as real as possible. Yeah, exactly. So for the humans, actually, for the heritability, you can still apply for the humans, because for most genes, it depends on the tissue. The median heritability could range from say 15% to 30-some%. So the median heritability is roughly say around 30%, right? But the kinship matrix actually depends on what data structure you are collecting. So if you are collecting a family study, especially if you are doing a twin study, then the kinship matrix is going to be much stronger than the Baboon we're using, because the Baboon are still, it's only involving relatively remotely related individuals. But if you say collect the human data, that's really a population scale study, and all individuals are supposed to be unrelated, then those have a much weaker, right, relatedness compared with the K-matrix and the Baboon. So the Baboon is kind of intermediate between all those various scenarios.
Hi, I was wondering if you tried this with any mouse experiments, because investigators are often very reluctant, understandably, to use multiple mice from the same litter because they want to avoid any kind of confounding with that. Would you advise people now with this method that it's okay to include multiple mice?
Yeah, so that's a very good point.
They're kind of wasting a lot of mice, by not using them from the same litter. And are there other things, if you did use it for that, are there other things that you need to take into account? Because they also have a much more similar environment being raised in the same cage.
Yeah, so that's a very good point. So in fact, my early works was motivated by some mouse studies. So the early works on linear mixed effects model in particular was motivated by this mouse study because now you have those related mouse inbreeding mouse, inbreeding mouse and mouse relatedness, and you always want to control for those relatedness. So that's why in early on in G1, especially in G1 studies, in the mouse population, people have to use the linear mixed effects model to control for those individual relatedness. And if you don't control for that, then you are going to see a huge signal. Even in the early linkage studies, you see a huge signal if you don't control for those mouse relatedness. So therefore, this method I'm developing here, the macaw, is particularly useful for those mouse studies because there, for this RNA sequencing studies in the mouse population, right, you can both account for its counter nature and you can also account for this mouse individual relatedness. So therefore, I believe this macaw model would be extremely useful. And in fact, we were trying to looking for some mouse data to demonstrate the effect of our methods, but we couldn't find some relatively large mouse data. So all we can find is like 3 mouse in one place versus 2 mouse in the other conditions. So those are extremely small data sets, and the power is extremely low, so it's hard to compare these different methods. So I think it's very useful. So if you know any good relatively large mouse data sets, please let me know. I'd be happy to apply to there. And for the second question, so that's a very good point. So basically the mouse, they are not only have a relatively large genetic relatedness, but many mouse, they are coming from the same cage, and there's a huge cage effect, right? Because in the cage, these mouse, they share the same food, they fight together, they eat together, and so share the same environment. So it's important, I think, to account for this cage effect. So in standard G1 studies, we do see a huge cage effect. And if you don't account for that, you still can have inflated type 1 errors. So I believe, I suspect it's the same thing for those RNA sequencing studies, and you want to control for the cage too. And there are 2 different ways you can control for cage. For example, you can label cage as a fixed effect, right? So cage 1, cage 2, cage 3, label it and include it as fixed effect, and that's easily done in the macaw. You can directly do that. And alternatively, you can model that as a random effect. So you can model using the cage labels as a covariance matrix and provide another covariance matrix there. And I think it can be done, although the computationally, you probably may not be able to use the macaw tricks over here. So the computation might be slower than the standard macaw algorithm. But that's a direction we're also looking at to try to see if we can improve computation there.
Okay, no other questions? Then well, thank you again, and thanks for coming.
Yeah, works with silicon.